Okay. So, good evening, everyone. Uh, Today, I will be taking the week 10 live session course on experimental biochemistry, which is being taught by Professor Shagota Dasgupta and Professor Shomode of IIT Kharagpur. So, welcome you all. So, I'm sharing my slides with you. So in today's class, we would be mostly discussing about the basics of recombinant DNA technology. So if we go from the basics, we have already seen and heard about DNA, that is the deoxyribonucleic acid. So DNA is a very large molecule made up of the nucleotides, and each nucleotide has three parts, mainly the sugar, phosphate, and the nitrogenous base. And these nitrogenous base are the materials that carries the genetic information. And you have these nitrogenous bases into purines and pyrimidines, among which you have the adenine, cytosine, guanine, and the thymine. So, what is a gene? So, the gene is a stretch of a DNA that codes for a type of a protein that functions for a particular organism. Okay, it is a unit of heredity in a living organism, and all living things depend on genes to carry their information to the upcoming generation. So genes holds the information to build and maintain an organism's cells and pass the genetic traits to the upcoming generation or the offsprings. So what are the components of a gene? So we have heard of exons and the introns. So exons are uh, the set of coding regions that codes for a particular protein, and you have the introns that are the non-coding regions or the removed sequence, and are called the split genes that are involved in the splicing of the exons to give us the entire construct, followed by protein synthesis of these exons, okay, into different proteins of our requirement. Now, what is the genome? So, the genome or the genetic complement of an organism, including all its genes as represented in DNA, is known as the genome. Now, there is a process known as gene expression. So, it's the process by which the information from a gene is used in the synthesis of a functional gene product, that is our protein of interest. Now, the process of gene expression is used by all known forms, like you have the eukaryotes, the prokaryotes, viruses, to generate the macromolecular machinery for life. Now, there are different gene expressions that we do. That is the transcription, followed by so the transcription is the mRNA synthesis. Then we have the post-translational modifications, post-translational processes, like you have the RNA splicing, then translation, that's the protein synthesis, and post-translational modification of protein. So the entire thing is going inside a cell. You see, you have the DNA to the mRNA via transcription, then the movement of the mRNA into the cytoplasm via the nuclear pore. You have the synthesis of the protein, and then the post-translational modifications. Now, what happens when these genes gets mutated or there is an alteration? So these leads to genetic changes. Therefore, an alteration in the segment of a DNA, which basically disturbs the gene's behavior and leads to disease. So it may be the small genetic change or the genetic drift mutation, or large genetic changes. That's the genetic shift, or we call it the recombination. So it's very important. So small genetic changes leads to genetic drifts. That's because of mutations, and recombination leads to a shift in the generation, or the genetic shift. Now, what is a mutation? You will see that there are several changes that can occur because of a mutation, or because of a change in the nucleotide sequence. Like you have the CTT, that the T, the middle thymine can get change to an adenine, and then you will see the coding mRNA sequence leads to a different kind of it will code for a different kind of an mRNA, and you will get a different kind of a protein. So one change in the nucleotide can lead to the formation of a different protein with a mutated protein sequence. Now from here you see that when the CTT gets changed to CAT from glutamine or a glutamic acid, you ultimately get valine, and ultimately this leads to a diseased condition, that is from a normal hemoglobin cell you get a sickle cell anemic cell. Okay, now that's all about mutation. Now the recombination is something that leads to the genetic shift. Now this is the exchange of corresponding DNA segments between the adjacent chromosomes during the special types of cell division that results in the production of new genetic makeup, and we call this as the genetic drift, the genetic shift or recombination. Now, the very basics about recombinant DNA starts where the DNA molecule that is formed in the laboratory of genetic recombination, such as molecular cloning, is used to bring together the genetic material from multiple sources, creating sequences that would otherwise have not been found in the genome or is not naturally occurring. Now, or the recombinant DNA is possible because DNA molecules from all organisms share the same chemical structure. They differ only in the nucleotide sequence and within the identical overall structure. Now, recombinant DNA is the general name for a piece of DNA that has been created by the combination of at least two strands. Now, recombinant DNA molecules are sometimes called the chimeric DNA because they are made up of materials from two different species, like the mythical chimera rDNA technology that the recombinant DNA technology uses. And using this technology, we can isolate and clone single copies of gene on a DNA segment to an indefinite number via a polymerase chain reaction, and these new combinations are introduced into the host cells that helps them to propagate and multiply and show up different functionality when incorporated in different organism, and that is what we want to catch. So overall, the sequence that was not naturally occurring has been genetically modified, are mutated, and that has been incorporated into a particular plasmid via the molecular cloning, and this plasmid is incorporated in an organism to check the function of this particular protein in that organism, or we want to check the expression of the genes and all. So this entire technology is known as the recombinant DNA technology, or RDT. Now, when we want to do recombinant DNA technology, or the basic step of recombinant DNA technology is molecular cloning, we need to know a few basic details. That's the restriction enzymes. So what are restriction enzymes? So these are bacterial enzymes that cut long strands of nucleotides into the smaller segments. It recognizes specific sequences, palindromic sequences, and cuts the DNA at specific sites within the sequence. Now, in one chain, the sequence runs from left to right, that is 5 prime to 3 prime, and from the other complementary sequence, it runs from the 3 prime to the 5 prime. But just because it's palindromic in nature, from which side ever you read, like it's GAATTC, and from when reading from the 5 prime to 3 prime, it is also GAATTC. So these restriction sites can leave like you over you here you have the sticky ends, but it can also have blunt ends when it cuts in the middle. So you can have GAACTT over here, and you have CTT over here, and GAA. So from whichever side you read, so the when you have the sticky ends, so these are the sticky ends, but when it cuts in the middle, so you will get the blunt ends, and they recognize palindromic sequences. So single chains or the tails of DNA are called the sticky ends, are created by the DNA segment by the action of restriction enzymes at a particular end, but when it does in the middle, we get blunt ends, and the sticky ends are readily binding to the complementary chains of the DNA. Next, we have the pieces of DNA that have been cut with the same restriction enzymes can bind together to form new sequences of nucleotides. And restriction enzymes can be used to isolate or cut out a specific gene. Now we have there are many types of vectors, like you have the cloning vector, and you have expression vectors. So cloning vectors, it's a DNA carrier used to clone a gene and transfer it from one organism to another. Now, once the gene has been isolated, that small piece of DNA can be placed into a cloning vector and can be introduced into an organism. Now, many bacteria contain a vector that we call as a plasmid. So, what's a plasmid? It's a ring of DNA found in bacteria in addition to its main chromosome. It can be used to transport genes from one organism to another. So, what's the process of using a cloning vector? So, we have a plasmid that is removed from the bacterial cell using a restriction enzyme is a cut and a donor. So, donor gene is a specific gene isolated from another and introduced into another plasmid. So, the plasmid is returned to the bacterial cell where it replicates as the bacterial cell divides. Now, this clones the donor gene. Now, the bacteria containing clones of the donor gene then can be used to infect other organisms and transfer the gene to them. In some cases, plasmids are used to clone a specific gene so that the bacteria will produce a specific protein. For example, insulin is a protein that controls the sugar metabolism. So you transfer the insulin gene and you get the recombinant plasmid. So you clone it into the bacterial in this plasmid and you transform it into the cell. You will see you get the insulin protein. So the different steps of obtaining recombinant DNA is that. So the step one is the DNA fragment containing the gene sequence to be cloned or the insert is isolated. Then you are cutting the DNA, then joining the DNA, insertion of these DNA fragments into the host cell using a vector. Now the rDNA molecules are generated when the vector self-replicates in the host cell. Transfer of the rDNA molecules into the appropriate host cell. Selection of the host cells carrying the rDNA molecule using a marker. Replication of these cells carrying the rDNA molecules to get genetically identical cells or the clone. So the first one is isolation. The first step in making the recombinant DNA is to isolate the donor and the vector DNA. Its use for obtaining vector DNA depends on the nature of the vector, and bacterial plasmids are commonly used vectors, and these plasmids must be purified away from the genomic DNA. Then you go for ultracentrifugation for extracting the plasmids DNA. That can be achieved by ultracentrifugation, and the plasmid's DNA forms distinct bands after ultracentrifugation in a density gradient medium. Now there is a protocol for extracting the plasmid's DNA can be achieved by ultracentrifugation, and this forms distinct bands in a density gradient containing ethidium bromide. The plasmid band is collected by punching a hole in the plastic centrifuge tube. And then you go for an alkaline lysis. So you will see that at specific alkaline pH the bacterial genomic DNA denatures, but plasmids do not, and upon neutralization the genomic DNA precipitates out, whereas the plasmid stays in the solution, and then you can isolate it using a silica column. Then after that you go for like restriction enzyme digestion, and that is cutting the DNA. So you can use different restriction enzymes for cutting the DNA and formation of the sticky ends. Okay, then you insert it into the vector, so you also have to digest the vector using that restriction enzyme, and you use for the insertion of the insert into the vector via the DNA ligase or via the process of ligation. Now after the ligation process is complete, then you have to transfer the DNA into the host cell. So you have the plasmid vector. So the vector is added to the flask containing the culture of E. coli, and calcium ions usually forms calcium chloride are added to the flask followed by the heat shock. So that's the process of transformation. Then you have different phage vectors that is introduced by infection of bacterial lawn growing on an agar plate. The culture or growth of viruses is made more difficult than the culture of bacteria or fungi that makes the virus grow. Now you are placing the gene in the vector, so you have the plasmid DNA. The DNA molecules are small and are easily separated based on size. So here the cells are broken up and the chromosomal DNA is centrifuged down. So you get only the plasmid DNA, and it's purified, cut with a restriction enzyme. Then you use a ligase buffer to ligate it. So that's the entire graphical thing. So then you go for transformation. So you take up a bacterial cell and make it competent using calcium chloride and some bigger sized ions, and you insert the plasmid onto it. So yes, so first you have to choose the bacterial host. That can be E. coli as a model organism because it's well studied. So it's well studied. It does not have nuclear membranes. It has enzymes necessary for replication, grows rapidly. That's 20 minutes is the generation time. And it's expensive, normally not pathogenic, and it's easy to work with and transform. The second step is bacterial transformation. You choose a plasmid to transform. Then the characteristics of a useful plasmid: single recognition site. It has the origin of replication to make replicate and make copies of the new cells. It should have marker genes, that's the antibiotic resistance, and the expresses a color if that's okay. Then you prepare the cells for transformation. As I was telling, you go treat it with calcium chloride that softens the bilayer and allows the plasmid to pass through, or you can do by electroporation, brief electric pulse, but not for mode, or directly inject the plasmid into the bacterial host. Then you provide it nutrients for the growth, incubated on agar plates at 37 degrees. That's the optimum temperature in which a bacteria grows, and you analyze the plates. Now, there is a very important thing that you need to know when you do cloning. That's the polymerase chain reaction. So, in the last class also I was telling. So, it was done in 1985. It's a very fast, powerful, adaptable, and simple technique. Now, why do we need to amplify the sequences? Because we need to use it for cloning and sequencing. So, we need more amount of DNA. We need to identify the engineered DNA constructs, then monitor gene expression, diagnose diseases, then reveal the presence of microorganisms and identify individuals. So for PCR, we need a template DNA that will act as a target sequence. We need primers, that are short oligonucleotides that will define the ends of the target sequence to be amplified. We need thermostable DNA polymerase, buffers, DNTPs, a thermal cycler. And then we have a PCR reaction in the first cycle. Okay, so this is how a simple PCR reaction works. So that's the denaturation by the heat followed by primer annealing. Then you have the DNA synthesis, primer extension. Then you have the primer extension followed by so first is the heat denaturation, then the primer annealing, and then the primer extension via the DNA polymerase. So each PCR reaction, so if you have a molecule of DNA, it will give you two DNA molecules after the first cycle, and at the end of 30 cycles, you get about around 10 to the power 9 molecules of DNA. Okay. So now we will discuss the practice problems. Okay, so let's move on to the first question. So plasmids can be successfully used as vectors for gene cloning as so they are extra chromosomal circular double-stranded DNA, or they possess selectable marker, or all of the above. So the properties of a gene cloning vector is that they should have all the three properties. Now to cut a DNA molecule, which of the following enzyme is used? Ligase enzymes does ligation. Restriction enzyme is the one that does the restriction or cuts the DNA. Phosphatase adds a phosphate group, and you have the polymerase that goes for the DNA extension. Now the DNA molecule to which gene of interest is inserted for transferring it into another organism. That is our converter, vector, exporter, transporter. That is our vector. Yeah. Now, to join gene of interest with the vector, which of the following enzyme is used? Ligase, lyase, ribonuclease, polymerase. So, as I was telling, to join the DNA, we need to use ligase enzymes, DNA ligase or something. So, ligase. Now, the mechanism of intake of DNA from surrounding environment by a cell is called conjugation, transduction, transformation, or gene cloning. So you're taking DNA from the surrounding environment. What is that? Yes, that's transformation. C, that is correct. Next one, what is the role of the selectable marker in cloning vector? Maintaining the vector in cells, selective survival of cells that have the vector, increasing the cell growth, expressing the gene in the cell. So which one? So what's a selectable marker? That's the antibiotic resistance. So it's selective survival for the cells of the vector, that is correct, as well as for maintaining the vector in the cells. Now you have an MCS, that is the multiple cloning site in a vector. So what does the MCS have? Promoter site, ligation sites, restriction sites, or the antibiotic resistance. So what does the MCS have? So what is multiple cloning sites? So those are the sites that have identifiable sequences by the restriction enzyme, that it can identify the palindromic sequences and cut at different portions. So you have different restriction enzymes identifying different sequences and cutting. So these are the unique restriction sites. Now question 8, that is what will happen if the origin of replication is missing in a vector. So if the origin of replication is missing, the vector will not take up by the cell, the vector will not contain the gene of interest, vector will not survive in the cell, or the vector will not replicate. Very interesting question. So if there is no, so the origin of replication, and from the name as we can suggest, so here the replication machinery starts, so the vector will not replicate. Okay, now next one. A large number of copies of a gene can be obtained following transformation, polymerase chain reaction, plasmid isolation, or the whole genome extraction. So, I just mentioned about the technique known as the polymerase chain reaction, or the PCR, that is used for the amplification of the genes. So, large copies of gene can be obtained by polymerase chain reaction. Question 10. Which of the following statements is true? PCR machines are known as thermocyclers. Exponential amplification of the gene can be done from a very small amount of DNA. Thermostable DNA polymerase is absolutely necessary, or all of the above. Yeah, so PCR machines are also known as thermocyclers because they modulate the temperature. Amplification of the gene can be done. That is correct. And thermostable DNA polymerase is absolutely necessary. So all the three are correct. The first step of PCR. So what is the first step of PCR? Renaturation, denaturation, annealing, and extension. So how does the PCR start? You add the template DNA, you add all the enzymes and the master mix, then you heat it at 98 degrees. That's the denaturation step. Then your primer anneals and extends. To which side of the primers the restriction sites are included for cloning? 5' and 3' end, both 5 and 3' ends are not present in the primers. So which side of the primers are included for cloning? That's the 5' end that marks the two boundaries. The stop codon is present in the 5' end of forward primer, 3' end of forward primer, 5' end of reverse primer, or 3' end of reverse primer. So what is the stop codon for? The stop codon is used for will be when the mRNA is made, it has to be in the 3 prime end. So when it is in the 3 prime end, so the stop codon has to be in the 3 prime end. So therefore it will be in the 5 prime end. That is, it has to be in the 5 prime end in the primer, but in the reverse primer, not in the forward primer, because the forward primer will code for the starting sequence. In PCR, the DNA synthesis takes place at, so at what temperature does DNA synthesis takes place? So the optimum temperature at which our DNA polymerase works is 72. So you heat it at 98 or 95, then you cool it down to an annealing temperature depending on the melting temperature of the primers, but extend it at 72 degrees. That is the optimum temperature at which a DNA polymerase works. The last question, expression vectors differ from cloning vectors by having selectable marker, gene expression control elements, origin of replication, or all of the above. So selectable marker is present in any vectors irrespective of whether it's an expression vector or a cloning vector. So selectable marker, gene expression control elements are, so selectable marker is present everywhere. The gene expression control elements, so that is the one that will be there, because cloning vectors cannot undergo gene expression. Origin of replication has to be there because all the vectors undergo replication. So that's all. Yeah, so that's all from the practice problems. Okay. So if you have any questions, you can ask me, else we can end it over here.